The overall goal of the following experiment is to screen and evaluate genes required for invasion and/or survival inside macrophage cells in vitro in a high-throughput fashion and identify mechanistic genes of Salmonella pathogenesis. This is achieved by adding wild-type or mutant Salmonella strains to macrophage cell cultures in 96 well plates for 30 minutes as an in vitro infection. As a second step, after the 30-minute incubation, Salmonella that have not attached to the macrophage cells are washed away and fresh medium containing a high concentration of gentamicin antibiotic is added for 1 hour, which kills all the extracellular bacteria that are not internalized. Next, the macrophage cell culture is washed again, and fresh medium containing a low concentration of gentamicin is added for an additional 22.5-hour incubation in order to maintain a bacteria-free extracellular environment and allow only the intracellular Salmonella to survive and proliferate. The results show that by comparing the mutant Salmonella with the wild-type strain, the in vitro phenotype of the mutant strains for association, invasion, and replication is determined based on the measurement of colony formation units per macrophage cells obtained by the serial dilution plating assay for each time point.
The main advantage of our technique is that it measures association, invasion, and replication of Salmonella in phagocytic cells, and that it can measure multiple strains simultaneously for higher throughput. Demonstrating the procedure will be Dr. Jing, postdoctoral research fellow in my laboratory, and Mrs. Roberta, Pugh, research associate in my laboratory.